All right, now that we've had our introduction into the different aspects of genetics and we just completed cell biology by going over the cell cycle, now we're finally ready to actually start the genetics part of the class. And so, if you recall from the overview, we're going to start with Mendelian genetics, which is what later became the very beginning of what we call classical genetics, or more importantly, what we call transmission genetics. So the trajectory of this course is going to be, we're going to start with our transmission genetics. We'll go over that. Then we'll do the cytogenetics. Then we'll get into the molecular genetics at the end of the course. So let's start into it. So now you're in a genetics course, so you knew you were going to hear about Johann Gregor Mendel, right? Johann Gregor Mendel is basically, he's known as the father of modern genetics. And it's really quite extraordinary what he was able to accomplish. So he was the first to really experimentally uncover the fundamental principles of genetics. When I say experimentally, remember there were lots of people that had all sorts of ideas of how we pass on our traits to our offspring. But of course, they didn't have any scientific data, scientific evidence, or anything like that. Gregor Mendel was an extremely meticulous scientist. So he was a brilliant statistician, and really his work it's to an untrained eye such as myself, I would question whether he was obsessive compulsive because it is impeccable work. So much so that there are many individuals that actually disagree that, or not disagree, that discount his work and think that he, fudged his data, or he basically was not of the highest scientific integrity, right? He was making data up or omitting negative data, that sort of thing. But again, I think I may have said this earlier in the course, you can repeat all these experiments. So if you can repeat them, then your argument as to whether he did or did not do his work seems to fall short. So I'm in the camp that believes he actually was able to accomplish all that he accomplished and was really as lucky as I will try to convey to you, which is absurdly lucky. So his use of pea plants rapidly sped up the study of inheritance. We just talked about how in the first portion of the course we talked about how you want to use certain organisms that provide you with, the ability to propagate them efficiently, cost effectively, and then most importantly, you want a decent number of offspring in a reasonable amount of time. And so by using pea plants, that, like I said, rapidly sped up the study of inheritance because, it takes over 20 years for us to get any information really about human inheritance. It's not just 9 months, you get a baby and then you figure it out. You obviously need to wait for the people to reach sexual maturity so they can reproduce. Then they have the child, and then it depends on what you're studying. You might need to wait for that child to reach sexual maturity or to reach an upper age, get up there in age. If you're measuring something, I think the example I used previously was like the graying of hair. So you have 20 plus years for humans, and your sample size. Remember, this is a brilliant statistician we're talking about. So your sample size is going to be what, n equals 1, maybe n equals 2. So you're not going to get very many offspring per generation. Whereas with pea plants, you can get as many offspring as you want because you plant seeds, and then more importantly, you have 100 days for pea plants to regenerate. So that's about 3.5 generations per year. That is an extremely reasonable time frame to work with because Mendel was planning out his experiments. Like I said, he was extremely meticulous. He spent years planning out his experiments before he actually performed any. And 100 days for pea plants. So that means that gives you enough time to count up your offspring and take your measurements and then sort of try to interpret your data before you're on to the next generation. Now, he also chose pea plants because like I said, he was thinking about his experiments for years. And so he went to local markets and he decided on pea plants because, so P. sativum is the genus and species that he decided upon. And he decided on them because they had a number of different traits that could be studied. Now, remember, we don't know anything. This is the 1800s. We don't know anything about genes or chromosomes or anything like that. We're just looking at traits that you can study. So actually things that you could see in this case with the pea plants. And so he noticed that they had a number of traits that could be studied. And remember, at the time, at Mendel's time, Um, he was a firm believer in the prevailing theory of inheritance of the time, which was blending. And so he just set out to scientifically uncover how blending occurred. And so one of the ways he could do that is by studying pea plants because they had a number of different traits that could be studied, including the fact that they were self-fertilizing. So first and foremost, forget about what you're looking for. The fact that they were self-fertilizing, it means that he could do a cross and cross two strains, or if he hasn't quite figured out what was going on, or he just wanted to keep a genetic stock alive, he wouldn't have to cross it with another strain just to get offspring so that he could keep it alive, like, when we breed animals or something like that. What he would do is he would just be able to self-fertilize. You could literally take pollen from one flower with a little glass stirring rod, and then you could go pollinate another flower on the same plant, and that would be self-fertilization. That would work. That would give you viable offspring generation after generation after generation. So you never have to worry about anything going sterile or anything like that. So they were self-fertilizing. He was also planning on doing massive experiments because, again, brilliant statistician. So he was going to do very large-scale experiments in terms of sample size. So he's going to have a lot of these pea plants growing in a little modest greenhouse that he had. And so that was going to be tight quarters. And so he noticed that they had a flower structure that minimized accidental pollination. Very important because while you're working with a cluster of these pea plants in their teeny tiny greenhouse, when you go to, like, say, for example, self-fertilize one, if your elbow like brushes up against a pea plant, like, a couple plants down the line, and then you move over and you're working on the next plant, and then your elbow brushes up against the next plant down the line, a couple plants ahead of where you're working, well, you don't have to worry about your elbow being a little honeybee and then pollinating flowers for you or anything like that. These flowers are not going to be pollinated unless you wanted them to be pollinated. It took a little effort to get into the flower structure was not going to allow for, like I said, accidental pollination, as you can see here. And that was very good for him because he thought, oh, okay, well, that means that I can keep track of my experiments and I won't have any, he's trying to basically minimize outliers, minimize any sort of mistakes that could be made that would throw off his statistical analysis of his data. And then, of course, I think I may have said this already, even if you self-fertilize or if you were to cross them, either way, their offspring were fully fertile. So that means that future crosses could be made. You could keep these genetic stocks alive generation after generation after generation, and then you can determine whether you want to whether you want to cross it or not cross it, or you just save it as your control or whatever that may be. All right. Now, I said that he was a brilliant statistician, and I said that he spent years setting up his experiments, or even planning for his experiments. We don't even know exactly how long he was thinking about these experiments. What we do know is that once he kind of started these experiments, and I say kind of because you'll see in a moment. So what he did was he went to different markets, he obtained pea plants, and then if you bought a pea plant from somebody, right? So, and the salesperson says that, oh, this pea plant that will give you yellow peas every year, guaranteed, right? And so he would just basically be like, oh, okay, thank you, I don't believe you, and so he went back to his greenhouse, and once he obtained his strains, he tested them. He tested all available varieties of peas that he had access to for 2 years, just to make sure that he had what he thought he had. So you buy a strain that only gives you yellow peas, and then you test it for basically 7, 8 generations before he was comfortable actually saying, okay, all right, this pea plant actually only gives you yellow peas, right? And it's not like the salesperson is standing over and going like, I told you so. But realistically, that's what he was doing. He was trying to make sure that he really, truly understood what he was working with. And so he basically came up with, or made sure, he ensured, after 2 years of testing, that he knew what he was starting with, and he called them true breeding lines. And so they're true breeding lines. What that means is that they are true breeding. Like, for example, this strain is true breeding for yellow peas. It will only give you yellow peas, yellow peas, yellow peas, yellow peas. It will always give you yellow peas, and so therefore it is true breeding for yellow peas. It's never going to switch to like green peas here and there. It is always going to give you yellow peas. That is a true breeding line. Some of you that have a little bit of background in genetics, you might actually know that what we're saying here, we're using Mendelian terms because we haven't even coined the terms that we use today yet. According, on Mendel's timeline, they didn't exist yet. But if this is a little confusing to you and you have a little bit of a genetics background, you learned a little bit already, then what I'm talking about is homozygous line. So it could be homozygous dominant, homozygous recessive. We'll go over what those mean a little bit later on, but for right now, let's call them what he called them, true breeding lines. And so here's your definition. True breeding lines are lines that give rise to the same traits in all offspring generation after generation after generation after generation, indefinitely. And so I mentioned he did massive experiments, right? And so he spent a lot of time working on this. He was a monk in the 1800s, so it's not like he had, internet and all sorts of other distractions. He was very focused on his work. And so he performed experiments on a large scale because, again, I can't say this enough, brilliant statistician. And so he had sample sizes of n equals 28,000. So he used 28,000 plants in one experiment just to avoid artifacts, to make sure that he avoided artifacts. So you can imagine there's a lot of plants that he crammed into a small little greenhouse, and he worked with them and took meticulous notes. So, therefore, he is that's why he gets to be called the grandfather of genetics, right? Or the father of classical genetics, I believe is what his unofficial title is. So his methodical, hypothesis-driven, and that's important because other people were just using the Socratic method and logic. He's doing hypothesis-driven research with experimentation, documentation, data, and data analysis. So his methodical, hypothesis-driven approach was the key to his success. That... And the more you learn about it, the just harder it is to fathom how lucky this guy was. So methodical, hypothesis-driven approach and extreme luck. He is the reason why there basically is a phrase. Actually, the phrase "chance favors the prepared mind," I believe, was Sir Alexander Fleming and penicillin in his discovery. But realistically, this guy was super lucky. There is no way he could have known so many different things. And so, of course, all sorts of people make all sorts of really repetitive and tired jokes about how he, was a monk, so he had help from above, et cetera, et cetera. But realistically, very lucky. But also, forget about luck. Not just the luck. The methodical, hypothesis-driven approach was also extremely important. So, Mendel studied 7 characters, as he called them, that affected the seeds. So, fun fact for you that you don't really, I'm never going to test you on this, but, actually, you might need to know, yellow peas are actually dominant to green. So we tend to like our green peas when we eat, right? We eat green peas, and so humankind seems to really focus on green peas. But if we didn't artificially select for the green peas, we'd all be eating yellow peas, because yellow peas are actually dominant to green peas. These are the kind of things that I, basically... happily tell my young daughters at the dinner table while I'm trying to get them to eat their peas, and it works because they basically want me to shut up, and so they eat their peas. So anyway, so here, I'm getting paid to tell you that because technically, that's genetic information there. So So yellow versus green, that's one thing you can look at. That's one thing he measured. Here are the 7 things that he measured, right? So seed shape, round versus wrinkled, seed coat color, gray versus white, pod color, pod shape. Here's a good one. So axial versus terminal, whether the flowers were along the stem or at the top, and then short versus tall. So here's the thing about the pea plants. It's very, very interesting. And actually, this is what led to the simplicity, the pseudoscience of eugenics. In this strain of pea plants, a lot of the things that Mendel looked at, these 7 characteristics, were very, very binary. Very just this or that, black or white. It is like we measure height not by saying, hey, there are short people and there are tall people. I actually use the term, we measure height because there's multiple overlapping categories that people fall into, right? So like if we break it down to the inch level, well then, okay, so there's the people that are 5'1", 5'2", 5'3", 5'4", 5, let's just say, like 5'10". Well, you could go further, right? In the 5'10" category, there are those that are like exactly 5'10". There are those that are like 5'10 and a half, right? Not quite 5'11", etc. And so but with pea plants, what's really interesting is that it turned out, I'm kind of jumping ahead here, but let me just say it. It turned out that there was one gene for each one of these 7 different traits, just one gene. And then there were only 2 versions of that gene, 2 alleles. And so you end up with this or that. And so that is why people thought, well, okay, there could be, in human beings, one gene for something very complicated. And you could either be this or that, right? You could either be like, if you have the boat builder gene, you could either be a boat builder or not. And so, unfortunately, that pseudoscience led to the eugenics, the movement and the things I talked about in a previous lecture that you can certainly access via Polylearn, or just on our course website, Canvas or Polylearn. And so that was, it was very interesting that it got extrapolated to human beings. But let's go back to what we were starting with. So now he hasn't even figured all that stuff out yet. So I got a little ahead of myself. So what we're looking at are 7 different characteristics. Notice these are things that you can easily just visualize because, again, he's just trying to see how things get passed on. And these are nice things to visualize because, look, this is short, this is tall. And so if blending were to occur, well, then we would have a pea plant, the offspring, that was like kind of in between, right? So it would be not short, not tall. It would be like medium height, right? It would be like, I don't know, shawl. And so, you have, yellow versus green. So he would expect it to be, so it would be somewhere between yellow and green. So let's go with green and yellow. So let's go with grello, right? Okay, I'll stop. Okay, so basically the genetic terminology that we use today, Mendel came up with some of it, but things like genes and chromosomes, those are terms that Mendel never knew, right? They were coined well after his death. And so he called inherited factors. We know them, I just mentioned on the previous slide, we know them as genes now, right? So a gene for seed coat color, a gene for, plant height, but he called them inherited factors. What he did come up with was a term we still use today, and that is alleles. So there are different flavors of what we call genes, right? He had different versions of his inherited factors, right? There was like yellow peas and green peas, and so he dubbed those different alleles. So alleles are different versions of an inherited factor, or what we now call a gene. I'll do my best to try and use Mendel's terminology until we introduce the terms, but realistically it's very difficult for me because obviously I've been using modern terminology my entire life. And so I mentioned P. sativum, so Pisum sativum is the genus and species of the pea plants that he used, and they have one gene for seed shape, and they have that could be either of 2 different alleles. So smooth versus wrinkled would be like, for example, the seed coat, right? Sometimes the peas would be smooth, sometimes they would be wrinkled, and so if you're working on trying to see how blending occurs, you would cross a smooth and a wrinkled, and you would expect something in between, and I can't help myself. You would expect something that's sminkled, right? And so well, no, I'm not going to stop. So basically, you would expect something in between. That's what he was trying to determine. How do the traits get passed on in such a way that they get blended? And the theory behind, remember, blending is they can't go back, right? So like once it's an intermediate phenotype, right, or sminkled. Once it's already mixed, once it's blended, you got something in between. Then you cross it again, and it should just keep getting diluted and changed, but it should never go back to the original. That's the prevailing theory of blending, of Mendel's time when he was working on this, when he first started working on this, and that's what he was set out to basically decipher with experimental data and data analysis and hypothesis-driven research. So just a quick little follow-up on an allele. All alleles of a particular gene will be found, well, we know this now, all alleles of a particular gene will be found at a specific place. All right, so you can see here's a chromosome, here's the centromere, this is the P arm, this is the Q arm, here's the gene for R, whatever R is, and then you could say, okay, well, usually the uppercase letter is reserved for the dominant allele, and then a lowercase letter is reserved for a recessive allele. It could be multiple letters and numbers, but the genes are usually italicized, and uppercase is dominant, lowercase is recessive. And of course, we haven't gone over what dominant and recessive is in the course yet, so that's coming up. But I should say all alleles of a particular gene will be found in the same specific place on a chromosome, and we call that place a locus. or there are two loci shown, right? So one on this chromosome, one on that chromosome. So loci is plural, locus is singular. All right, so let's start into his first experiment. Years of setting up for this experiment, right? So he's setting up his first set of experiments. And so what he's going to do is try and, so very, very, I've said brilliant statistician so many times, we could actually do a statistical analysis of that. But he's not only a brilliant statistician. To be a brilliant statistician, you have to have a pretty decent basis in mathematics. And so his first job was to figure out how blending occurs. And so what he wanted to do was minimize his variables. You have 7 different things that you're looking at. You don't want to do all 7 at the same time. You want to minimize your variables. So you're going to keep 6 of them the same. 6 of the traits are going to be exactly the same between the plants that we're going to cross. And 1 is going to be different. That would describe his first cross, which was he first performed what's called a monohybrid cross. Monohybrid crosses are those that basically are going to keep all the other traits the same except for the one. That's where the mono comes from. And then we're going to hybridize. We're going to cross of that one trait different alleles, different versions of that trait. So like, for example, a tall and a short, or a smooth and a wrinkled, or a yellow and a green pea producing plant. So yellow pea producing plant and green pea producing plant, or round pea producing plant, or smooth. Round and smooth are used interchangeably. So, like a smooth seed producing plant and a wrinkled seed producing plant. That would be if all these 6 of the 7, if the 6 other characteristics, they're both tall, they both have green pea pods, that sort of stuff. They both have axial flowers. If all that's kept the same, then you're just checking one thing. That is what constitutes a monohybrid cross. And so what he did was, we're going to look at, I can't help myself. We're going to definitely see if how you get sminkled. So basically, he crossed true breeding lines, right? So this is the P1 cross. Let's talk about a little bit of genetic terminology. P1 cross is P1 for the first generation and P for parental. So he crossed plants with that produce smooth seeds with plants that produced wrinkled seeds. And so this is your parental generation, your P1 cross. And he was trying to determine how they pass on their traits to give you something in between, an intermediate outcome, which is called an intermediate phenotype. And so he's expecting to get some offspring, the plants, they're going to grow up and they're going to produce peas that are going to be not quite as smooth as the smooth parent, not quite as wrinkled as the wrinkled parent, something in between, because we're going to blend them. And that's why I keep, trying to mix the words, blend the words to make, something silly that sounds, actually personally, I don't think it's silly. I think it's awesome. Sminkled. So the seeds, basically from this cross are your first filial generation. That's just a derivative. That's basically just a, I can't remember if it's Latin word root for family, right? So your first familial generation or first filial generation is the F1s. And here's what he saw, actually. What was really odd was that they were all smooth. So he did this on a massive scale. Right. So at first he thought, oh, I did something wrong. What did I do wrong? So he did it over and over and over again. And they were all smooth. And, this is a brilliant statistician. And so it really threw him off when they were 100% without any deviation, no outliers, 100% of the offspring of all of these plants for which he did monohybrid crosses would give you one. So you could see 100% would give you one of the 2 parental, traits, not the other. And so he did this for other, he has 7 traits that he's looking at. He did monohybrid crosses for all 7, and every single one, 100% of the F1 generation gave you the same result, which was similar to one but not the other parental, trait. And then there was no blending. Right, so he was like, "What is going on?" Because remember, he was trying to figure out blending. He believed in blending. It was widely accepted at the time, and so he was just beside himself. So what he did was he basically had to figure out what was going on. So he didn't do any more crosses at that point. He basically just, self-fertilized. 100 days goes by. He still can't figure out what the hell's going on. He's trying to figure out where he made mistakes and do extra crosses and everything. But every single time he's getting, 100% of the offspring are going to produce one of the two parental traits. And so, time goes by, and so he has to keep these strains alive because he's still trying to figure out what happened. So he's going to basically plant the seeds from the F1 generation. Basically, I should say he's going to self-fertilize the F1 generation so that it produces seeds, and then plant those seeds, and then keep the strain growing. And then, of the 7,324 seeds that he collected from one experiment, can you imagine that? Could you imagine picking pea pods, opening them up, and carefully cataloging every single pea that you get from a number of plants, such that you catalog 7,324 peas? And that's just one experiment. That is some commitment. Right. And so anyway, and so here's what he saw. And here is something that, if you saw this and you truly understood what was happening from Mendel's point of view, you would realize that this was like a tremendous plot twist. Right. This was that moment where like the audience gasps because, take that first monohybrid cross. He plants these. He F2 generation, they grow up, they start producing the seeds of their own, and then he notices that they are producing, the plants, some of them are giving you smooth seeds, some of them are giving you wrinkled seeds. So this is the moment at which he didn't recognize this as the moment yet because he wanted to make sure that what he was witnessing was not a mistake. But this is actually the moment at which blending, the theory of blending, dies. And so he has now disproved blending. And so you might be wondering, well, wait, hold on. This is just one experiment. That doesn't truly prove or disprove anything. I mean, we're talking about something on the order of like, there was a time when humankind thought the Earth was flat, right? And then everybody except Kyrie Irving realizes that the Earth is no longer flat, right? It was never flat. I'm sorry, let me rephrase that. We no longer believe the Earth is flat because we have all sorts of evidence of various sorts, including people that went up into space and looked back down at the Earth. And so the Earth is, roughly spherical. It's not perfectly spherical because it's spinning, and so it kind of bows out at the equator. But it's roughly spherical, right? And so we say the Earth is round. And so you know how entrenched in society that is now. Right, aside from like a couple wackos, right? There's a couple of people that just refuse to believe in any sort of scientific evidence, and so I'll just stop. So aside from them, it's pretty well entrenched in human culture now that the Earth is round, right? Well, at that time, it was pretty well entrenched in human culture that inheritance was through blending. It was so entrenched in human culture at that time that even now people still believe in blending. Many people believe in blending. If they don't understand science, if they don't understand genetics, blending seems like a logical option and they take that option. But back in the 1800s, Mendel was able with one experiment to disprove blending because here's the thing, blending, I think I used this example before, right? If you had like, say, blue paint and yellow paint and you mixed them, you would get green paint. You'd get something new. You would never have your green paint separate back out into blue and yellow paint. That's not going to happen. So the other thing that makes this a very powerful result is that in 100% of the F1s, you got one of the two parental outcomes. And then if you look closely at the numbers and you actually analyze your data here, this works out almost perfectly, and in every single monohybrid cross he did, it worked out almost perfectly, that you would get a 3-to-1 ratio of whatever was in the F1 comes back in the F2 in 75% of the offspring. And then whatever disappeared in the F1 comes back fully intact in 25% of the offspring. So, what's the quote, right? Men lie, women lie, numbers don't. And so, so Mendel realized that regardless of what he thought he made a mistake and was doing wrong, there is some sort of mathematical, formula at work here. And so you're getting consistent numbers. The 100% is no longer as scary in the F1 because it then basically separates out into 75 and 25%. And so at this point, Mendel realizes that he might be onto something. And so he is now intrigued because he now realizes that maybe, it took him a while to get to this point, right? He repeated his experiments quite a bit, and actually that was good because I basically just gathered more and more data, right? He was just building up his sample sizes. But it took him a while to really truly trust his data. And then once he looked at his data and started analyzing it, he trusted it, and he realized that he might actually be onto something, which is that blending is not how we pass on our traits. And so this is, like I said, the moment at which blending dies and actual Mendelian inheritance is born. So, like I said, this is a 3-to-1 ratio. A 3-to-1 ratio of, in this case, smooth versus wrinkled, but then he saw that 3-to-1 ratio in a number, all of his monohybrid crosses. The F2s show the 3-to-1 ratio. All right, so you could kind of summarize this set of experiments as follows, right? So the P1 is smooth versus wrinkled. The F1, you get all smooth. And then the F2, you get basically 75% smooth and 25% wrinkled. So you get a 3-to-1 ratio in the smooth to wrinkled in the F2s. And that's important. And so, like I said, he kept meticulous notes, right? Here's just a page from his notebook, right? People like me are like, wow. But basically, he kept meticulous notes. And the other thing is he did all of his analysis mathematically. So it's not like, Punnett wasn't even born yet. So it's not like we're using Punnett squares or anything like that. Okay, so what I mentioned was that results concerning all 7 characters were the same. So monohybrid cross, you get 3-to-1 ratio, 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 etc. So the F1 in all of these monohybrid crosses showed only one of the 2 parental traits. So 100% of the F1 were of one particular trait. It didn't matter which plant supplied the pollen. So if you think of the pollen as the sperm, right? The pollen is the male genetic donation from the plant. The flower is the female sex organ of the plant. And so, actually that'd be the pistil, but and so basically it didn't matter if you took the pollen from, you're doing a monohybrid cross. So you're crossing 2 separate plants, right? So like remember smooth versus wrinkled, you're a smooth seed producing plant and a wrinkled seed producing plant. So it didn't matter if you took pollen from the smooth seed producing plant and pollinated flowers on the wrinkled seed producing plant to do your cross, or if you took pollen on the wrinkled seed producing plant and pollinated flowers on the smooth seed producing plant to do your cross. When you reverse what you first did, right? Let's say we did pollen flower, and then when you reverse that and you do pollen flower, that's what's called a reciprocal cross, right? That's basically just, changing the genetic donors, making, the male genetic donor the female genetic donor, making the female genetic donor the male genetic donor. And so reciprocal crosses gave you the exact same results. The F1, 100% of the F1 were the same, right? So like in the case of, it didn't matter if you do a reciprocal cross, the F1s are going to be 100% smooth seed producing plants. And then in the F2, you could see that you got the 3-to-1 ratio. So the trait not present in the F1 reappeared in 25% of the F2 offspring. And so Mendel showed that these traits must therefore not be blended. They must remain unchanged because, you know what's happening, right? The wrinkled one, it basically did not appear in the F1 and then reappeared in 25% of the F2. That's a fancy genetic way of saying it skipped a generation. Right? The programming for it must have been in there, but just not being used in the F1, and then it reappears in the F2, in 25% of the F2. And so Mendel showed that traits remained unchanged as they were passed from parent to offspring. They were not blended, so they were individual units of heredity, right? And you know the definition of a unit of heredity, right? One definition of a gene is a unit of heredity. Well, Mendel is starting to realize that there are these self-contained units of heredity, these programs that are being passed on and not being manipulated or changed in any fashion. So they might not be expressed, But those, like for example, the programming to make a wrinkled seed was still present and passed on. So the F1s, those smooth F1s, still had it. They just weren't using it. And so the traits were inherited as separate units, and they don't blend. So he's realizing that blending is not how we pass on our traits. And so from these experiments, Mendel was able to conclude that each parent makes an equal contribution to the genetic makeup of the offspring. What he did was he basically had to make his numbers work. And the only way you can make your numbers work is if you realize that a 3-to-1 ratio means that each parent makes an equal contribution to the genetic makeup of the offspring, because that's how you can figure out the numbers. That's how he figured out how these numbers came about. And so he also made several conclusions that hold up even today. The first one is that genes, which he called inherited factors, determine traits, and they can be hidden or unexpressed. Again, the information encoding a wrinkled seed-producing plant is in here. It's just not being used or expressed. And so it skips a generation. Here it's back, right? It's being used in some of these offspring. And so, like, for example, F1 seeds contain a gene for wrinkled that was present but not expressed, right? Just what I said. And they reappeared in the F2 generation, the wrinkled seed ones. So he called the trait not expressed in the F1, but then comes back in 25% of the F2, a recessive trait. So there you go. Now we have our first definition of what a recessive trait or recessive allele is. So let me give you a little bit more information about what's recessive. So this is a true breeding line. This is a true breeding line. And so you'll see in a moment that Mendel basically figured out that you had to have 2 copies of each inherited factor in order for the numbers to work out. And so you would pass on one of those 2 copies. And so this is going to pass on the information to make a smooth seed producing plant. This is going to pass on the information to make a wrinkled seed producing plant. Now, excuse me, excuse me. All right, now, if they both passed on information, but then only one of them is what you see, well, then that one is masking the other one. And so we would say that the information to produce a smooth seed producing plant is masking the information to produce a wrinkled seed producing plant. And then it can come back in the F2s, because remember we have to self-fertilize, so we have to cross this with itself. And so sometimes you could end up with 2 instances of the smooth seed producing plant, two programs, or alleles of genes, right? Sometimes you could end up with, just like what you have here, 1 of smooth, 1 of wrinkled, and you still get smooth. And so then, of course, sometimes you could get wrinkled and wrinkled. And when that happens, you're back to a true breeding wrinkled seed producing plant. And so to describe all of this, you have to be able to describe the wrinkled seed producing plant's allele, and so that's the recessive trait. And then the one that masks it when they're present 1-to-1, the one that kind of covers it up, would be the dominant trait. So he called the trait seen in the F1 the dominant trait. The dominant trait is the one that masks the recessive trait. And so he called this phenomenon dominance. So we call it even today dominance or Mendelian dominance, right? So the reason why it's important that you realize that it's called dominance is because as we progress, right, we're going to go past what Mendel figured out. He didn't figure out everything. And so there are other variations of dominance. So we call dominance either straight up dominance or Mendelian dominance. And then not today, not in this lecture, but later on, we will get into codominance or incomplete dominance, other forms. So that's actually later on in this material. So we'll get to those. But right now we're working on Mendelian dominance, the very beginning of genetics, transmission genetics. So dominance led Mendel to conclude that while they looked identical, so that looks identical to that, the P1 and the F1 plants differed genetically, right? Those two differ genetically. So we need to have a name for this one versus this one. And so you can see they're right up here, up top here. And so it's important to make a distinction between an organism's appearance and its genetic makeup. So I've used these terms previously, but here they are in bullet points. They're official now. So genotype is going to be the specific genetic constitution of an organism. So this is true breeding for smooth. So let's call that, let's just use one letter abbreviation. So big S, because smooth is dominant. So big S, big S. Whereas this one is different. It codes, it looks the same, but its genetic constitution is different because it is big S for the smooth allele, and then we would say, okay, well, this is not smooth, right? And since this is dominant, that's what gets the lettering. So this is big S, little s. So big S for the smooth and little s for wrinkled. So this is big S, this is little s, and this is big S, little s. And so that has a different genetic constitution, a different genotype than this one. They have the same appearance or phenotype, but they have different genetic constitutions, so they have different genotypes. So phenotype is going to be the observable properties of an organism, and it doesn't have to be the stuff that you can actually see. So phenotype is commonly associated with, the appearance, right? So like in human beings, eye color, hair color, height, weight, that sort of stuff. But phenotype could also be whether you are, I don't know, whether you are lactose intolerant, right? You can't see that. Or what blood type you are. That's your phenotype in terms of your blood type. And so that will be dictated by your genotype. Your genotype encodes the information, and your phenotype is the final outcome, if you will, the observable outcome or observable properties of an organism. So, which of these have the same phenotype? Right? Well, we know that these two have the same phenotype. They appear the same. These have the same phenotype. And then here's the interesting thing. So, wrinkled and wrinkled have the same phenotype. And because they are recessive, they can only exist in the absence of a big S. This must be little s, this must be little s, so they also have the same genotype. So the P1 and the F1 and the F1 have the same phenotype. And then the P1 and the wrinkled, so the wrinkled P1 and the wrinkled F2s have the same phenotype, and we could actually logically infer that they also have the same genotype. So now I said, in order to make his numbers work, for him to understand the numbers that he was getting, his data, he already concluded that male and female parents contribute equally to the traits of the offspring. And so he figured out the simplest explanation, right? It's Occam's razor. The simplest explanation is likely to be true. And so he mathematically determined the simplest explanation is that there are two alleles for the seed-shaped gene in each organism. And so let's, like I said out loud, we'll dub smooth a capital S. So it's dominant, so it's capital S. So big S, for short. And then wrinkled is going to be little s. And so he noted that the gene number can't double every generation, so Mendel actually came up with, this is the beginning, this is the first human being to actually come up with the idea of meiosis, right? And he didn't call it meiosis, and nobody actually even knew about mitosis or meiosis for at least, I think, a decade after his death that we actually saw it under a microscope. Remember, this is the this is the mid-1800s, and so while microscopy has been around for quite a while at this point, it's been a hobby of the ultra wealthy, and so it wasn't a common scientific tool. Basically, Mendel was doing all of this by just looking at pea plants, writing down observations, and just analyzing his data. And so what he thought, or what he figured out, was that there was no way to get the numbers he was getting if the gene numbers were to double every time. So if they had two copies, if each strain had two copies, they didn't pass two copies on to their offspring. They passed one of their two copies. And if it was a true breeding smooth seed-producing plant, then it was big S, big S. If it was a true breeding wrinkled seed-producing plant, then it was homozygous recessive. So homozygous dominant, homozygous recessive are the genetic terms we use now. They'll be defined on pretty much the next slide. But basically, big S, little s, and so they each are going to give up one gamete. right? And so gametes are sperm cells, egg cells. In the case of plants, you have pollen instead of sperm cells. And so you have one. So you pick one of these, which you're going to get a big S. You pick one of, you pick one of these, you're going to get a little s, and that's what's ending up in your offspring. And that's why 100% of his offspring, the F1 generation, were always one thing and not the other. They were always showing the dominant allele as their phenotype, and basically the dominant allele was masking the recessive allele, so that didn't contribute to the phenotype. So he came up with the idea that gene pairs must segregate during this replication process or reproduction process. So that brings us to Mendel's first law, which is the principle of segregation. He came up with what's the reason why we just went over the cell cycle and mitosis and meiosis is so that you can appreciate that he was logically deciphering what later, many, many years later, decades later, was determined to be what we now know as meiosis and mitosis. So he came up with his first law of inheritance, which is the principle of segregation that states that the separation of a gene pair must occur during gamete formation. And so here's what it looks like. Now, this is the F1 cross. So when you self-fertilize, remember he was just self-fertilizing his F1s because he had no idea why he didn't see any blending in that first generation. And so he self-fertilized these, and he was later able to figure out why he was getting a 3-to-1 ratio. Because when you self-fertilize these, you can see that, okay, well, we're going to get one big S, one little s, one big S, one little s from each parent. And then there's randomization here. We don't know which two are going to end up together. And so we could see, using a Punnett square, that 25% are going to be big S, which would give you a smooth outcome. 25% are going to be big S, little s, which would give you a smooth outcome. Actually, another 25%, so that would be 50%, that are going to be a big S, little s, right? Because that's going to give you a smooth outcome. So that's smooth, smooth, smooth. And then finally, little s could occur in 25% of the outcomes. And so therefore, you would see smooth, smooth, smooth. So that's 3 to 1 wrinkled, right? Or 75% to 25% smooth to wrinkled, or dominant to recessive. So you can see that here, smooth, 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 wrinkled. And so there's where the 3/4 to 1/4 of the ratio came from. The 3-to-1 ratio is a phenotypic ratio. 3-to-1 is what they look like. You can see it's a phenotypic ratio. And if we wanted to say what the genotypic ratio is, we would say 1 to 2-to-1. But remember, the crazy thing is that Mendel figured all of this out. This is stuff that you know. You're like, oh yeah, that makes sense. That's meiosis. We just went over that. But this is all stuff that Mendel figured out before the discovery of any aspect of meiosis, mitosis, or even chromosomes for that matter. So Walter Sutton, Walter Fleming, right? So they're the ones, Walter Fleming is the one who identified chromosomes, and he didn't identify chromosomes until the early 1900s. And then Walter Sutton is the one who related what he was seeing with the cell cycle to Mendel's work. So Mendel did all of that without actually knowing about chromosomes or mitosis or meiosis. But the nice thing is that he was able to basically come up with a theory for how these pea plants were passing on their traits. And so his reasoning allowed the prediction of the F2 genotypes. Now, here's where you have a perfect example of how some people just don't trust his work. So what he did was he basically said, okay, well, now that I know how this works, I can set up crosses and I can predict how many of the offspring are going to be of a certain genotype, of a certain phenotype. And so if you look back on his work, it looks like he's literally saying, okay, now I am going to write down 7. 7, look at that. It's exactly as I thought, right? It's basically, it seems like he's kind of what's an analogy for it? Leading the witness, right? Seems like he's generating data to fit the observations that he's expecting. But in fact, what he did do was he designed the experiment, performed the experiment, gathered the data, and then he checked to make sure that that data did correlate with his theories, and in fact, it did. So here's what that looks like. So basically, he's now saying, okay, well, if my true breeding smooth seed producing plant is big S, and the wrinkled seed producing plant is little s, then my F1s are going to be big S, little s. And so therefore, instead of a 3-to-1 ratio, I can show that it is 25% big S, 25% little s, and then the remaining 50% is going to be just like the F1s. And the way he did this was, he didn't have, he couldn't sequence the DNA or anything. So what he did was he basically just kept, self-fertilizing after that first cross. So this is, a cross at the P1 generation, and then this is, okay, self-fertilization to give you these, and then just keep self-fertilizing each one of these and seeing what they produced. So Mendel confirmed these predictions through 5 successive generations after self-fertilizing. And so he was able to show that, so the 3-to-1 ratio was initially what he observed. But because he was able to reason and figure out what was going on and come up with the idea of 2 alleles, et cetera, what he was able to do was say, okay, well, then I'm going to predict that it's actually not a 3-to-1 ratio. It's actually, like we said a moment ago, a 1 to 2 to, so 1 to 2 to 1 ratio, or 25%, 50%, 25%. And so he was able to demonstrate that by just self-fertilizing these and getting the same results over and over again. So Mendel accurately identified alternative forms of a gene, or alleles, which are different flavors of genes. And he gave us some terms, right? So organisms with identical alleles are homozygous. So that's homozygous dominant. That's homozygous recessive, homozygous dominant, homozygous recessive. And then you would never say that this one is dominant or recessive because it's got one of each. So this one's heterozygous. So heterozygous. I think that's it. So basically, he gave us those terms. So homozygous. Homozygous, you usually want to say whether it's homozygous dominant, homozygous recessive. But heterozygous, you just say heterozygous. Okay. That brings us to our next step, right? So Mendel's like, okay, all right, now I have an idea of how this is occurring. Blending, he's left blending in the dust. Now he's voraciously trying to figure out, well, okay, he figured out what these traits are. Well, okay, do they interact with one another, right? So the next question is looking at 2 traits, not a monohybrid cross, but a dihybrid cross. So he next examined a pair of differing traits. And so he knew that smooth was dominant to wrinkled and that yellow was dominant to green. So he performed the following cross. So basically what this is a dihybrid cross. This is where you keep 5 of the 7 characteristics identical, but you change 2 of them. And so what he did was he crossed a smooth yellow seed producing plant with a wrinkled green seed producing plant to try and determine whether these inherited factors, or genes as you know them, the gene for seed shape, right, smooth versus wrinkled, had any relationship to the gene for the seed color. Like, could only smooth seeds be yellow, and could only wrinkled seeds be green, or are they independent of one another, or are they somehow linked to one another? That's the question. And so he performed a dihybrid cross. So what would you expect in the F1 generation if you performed a dihybrid cross such as this, right? Well, let's be realistic. The only S allele that this one can give is a capital S. The only Y allele that this one gives is a capital Y. The only S allele that this one can give is a lowercase s. The only Y allele is a lowercase y. So you're going to end up with big S, big Y, little y. And so you end up with a smooth yellow phenotype, but the F1 generation is going to be heterozygous for both. It's going to be big S, big Y, little y. At least this is what you would predict in the F1 generation for his dihybrid cross based off of the information we have thus far. The fact that smooth is dominant to wrinkled and that yellow is dominant to green. If they have no interaction with one another, well then smooth should still be dominant to yellow and wrinkled still should, I'm sorry, let me try that again. Smooth should be dominant, yeah, I said that right. Smooth should be dominant to yellow and wrinkled should be dominant. I did it. I see what I'm doing there. So smooth should be dominant to wrinkled and yellow should be dominant to green. I apologize. So now in the F2 generation, what do you expect? Well, he saw four different phenotypes. And so four different phenotypes tells you something, right? So first off, you have he saw smooth yellow, he saw wrinkled yellow, he saw smooth green, he saw wrinkled green. And then there you can also see the numbers here, right? So he saw a 9-3-3-1 ratio. And so well, forget the 9-3-3-1 ratio for the moment. Let's look at these individual numbers first. So he counted up 315 that were smooth and yellow, 108 that were smooth and green, 101 that were wrinkled and yellow, 32 that were wrinkled and green. And so the fact that, like I said a moment ago, the fact that you saw four different phenotypes tells you, okay, well, it seems like any combination is possible. And because all combinations are shown, right? And so that makes you think, okay, well, smooth's going to be dominant to wrinkled, green's going to be dominant to, yellow's going to be dominant to green. And so, excuse me. And so you can actually see, well, let's see if that holds up. Do we still see a 3-to-1 ratio? Well, here, let's count. So smooth, so we have what, 423 that are smooth. And let's see, where are our wrinkled? So our wrinkled, so we have 133 that are wrinkled. Well, I mean, if it was, I hesitate to say this, if it was 420 and 140, then that would be a perfect 3-to-1 ratio. But you're just pretty close. So you got a 3-to-1 smooth to wrinkled ratio. Let's count up the yellows. So yellow is going to be 315 plus 101, so 416. And green is going to be 108 and 32. So 140, right? So I think I misspoke a moment ago. 420 and 140 would be a perfect 3-to-1 ratio. And so in this case, we almost have a perfect 3-to-1 ratio between the yellow versus green. And so it looks like your 3-to-1 ratios are kept intact, which means that it seems like the individual, like the smooth versus wrinkled, yellow versus green crosses are happening regardless of the fact that there's another cross occurring at the same time. So whether it's monohybrid or dihybrid, that doesn't seem to matter. And so they seem to be independent of one another. Now, does anybody else, or I shouldn't say else, does anybody see anything else in these numbers? It's very important. Can you see it? Usually at this point in the lecture, most of my students can't see it. And so I have to tell them they're looking for smooth versus wrinkled or yellow versus green. But what you should realize is that we have been dealing exclusively with, well, first off, we have a 3-to-1 ratio that's still present for each trait. But here's our 423 smooth, 133. There it is. We're dealing exclusively with Cal Poly colors. Isn't that awesome? So that's it. There's nothing else to look at. It's just Cal Poly colors. I'm messing with you. So what we've got is, basically 420 to 140. That's going to be, so 416 obviously, but if it was 420 to 140, that would be a perfect 3-to-1 ratio. So realistically, your 3-to-1 ratios are still intact for each trait. When then you can look at, okay, well, these numbers overall, I jumped straight to these, but those are important. Now you see a 9-3-3-1 ratio overall. Just like with a monohybrid cross, you should associate that with a 3-to-1 phenotypic ratio. A dihybrid cross, you should associate that with a 9-3-3-1 phenotypic ratio. All right, so this led Mendel to come up with his second law, which is the fact that the smooth versus wrinkled has no effect on green or yellow versus green. It meant that he thought that they were independent of one another. So he came up with the principle of independent assortment. The principle of independent assortment is that alleles are distributed into gametes randomly during meiosis. That should sound familiar because that's the same thing we saw with the shuffling of the genetic deck when we were talking about meiosis. Right? So there was homologous crossing over, right? So that was the crossover events, that was homologous recombination, that was one thing. And then there was independent assortment or random assortment, either one. And so Mendel came up with that. Again, decades before we actually saw and documented it with chromosomes in meiosis. So his principle of independent assortment explains the inheritance of two traits, and that's how you get the 9-3-3-1 ratio. He assumed that alleles of one gene pair segregate into gametes independently of alleles of the other gene pair, meaning it doesn't matter which smooth or like big S or little s, whichever one gets into the gamete, that has no bearing on which version of Y you're going to get. Are you going to get big Y or little y? Doesn't matter. You can get a big S and a big Y. You can get a big S and a little y. You can get a little s and a big Y. You can get a little s and a little y. Any one of those are possible. So the resulting gametes have all combinations of alleles. And Mendel used probabilities to deduce this. So he's just counting up what he saw and then multiplying and getting out, oh, well, 9 out of 16 of the offspring are smooth yellow, 3, 3, 1. So 9, 3, 3, 1. Those are the combined probabilities, 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16. And so we just reduce that down to a 9, 3, 3, 1 ratio. Now, the principle of independent assortment explains the inheritance of two traits. And so we know from the F1 generation that all of them are going to be smooth yellow, right? The F1s, so here's the P1 cross, you're going to get all smooth yellow, right? But then, so they're all smooth, they're all going to be yellow. So here's the P1 cross again. There it is. They're all going to be heterozygous for yellow, and they're heterozygous for smooth, but they are going to be smooth and yellow. Now, the only possible outcome for the P1 cross, as I just said, is, smooth yellow. But when the F1s are crossed, there are 4 phenotypic combinations now possible, right? So it's kind of like FOIL. You're going to basically say, okay, well, I could take this S and I could pair it with this Y, or I could take this S and pair it with this Y, or I could take this S and pair it with this Y, or I could take this S and pair it with this Y. And so there is basically what you want to do is you want to be able to come up with the Punnett square, 16 square Punnett squares you're looking at right now, for a dihybrid cross. If you can do this, so on the next exam, you should expect questions about dihybrid crosses. And so if you can do this on paper from scratch with your any average dihybrid cross, then you will be in very good shape for any questions I might ask you about Mendelian genetics or inheritance. So let's go through it. So where does the 9-3-3-1 ratio come from? Well, okay, this is how you would set up this Punnett square. I just did, right? So big S, big Y, big S, little y, big Y, little s, little y. So those are the different combinations that are possible according to Mendel's second law. Well, if this is a self-fertilization, well, those are the same gametes that are going to be from the other, the same parent, but basically those are the gametes. So here you go. And now we can see where, so this is 16 squares. So remember it was 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16. Well, this is how we can calculate the outcome, right? This is how we come up with the 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16, or the 9-3-3-1 ratio phenotypically. Genotypically, you're going to get a lot of different stuff. But this Punnett square is really useful because we could see what is going to be smooth and yellow, smooth and wrinkled. Let me try that again. Smooth and yellow, wrinkled yellow, smooth green, or wrinkled green. So let's look at the first row. Right, in the first row, you've got a gamete that has a big S and a big Y in it. So really, it doesn't matter what the other alleles are in the other gamete. You're going to end up with something that's going to be smooth and yellow. Right? And so it turns out that the first one's actually, that's homozygous dominant for both. But it's smooth and yellow, smooth and yellow, smooth and yellow, smooth and yellow. Actually, the same thing goes for the first column, right? So you can see that that's smooth and yellow. And so everything here is going to be smooth and yellow. And so that gives us 1, 2, 3, 4, 5, 6, 7 that are smooth and yellow. Are there any other smooth and yellow outcomes? Well, if we look here, we have a smooth. So everything in this column is going to be smooth, but this isn't going to make it yellow. But if we go down, we already got this one, right? So where's here's a big Y. So that big S and that big Y tells me right here I should expect another smooth and yellow. In fact, that big S and that big Y tell me right here I should expect another smooth and yellow. And now we have the 9 smooth yellow of our 9-3-3-1 ratio. Here we have a smooth And it's going to be, well, it's going to be green, right? Because little y and little y means green. So that's going to be smooth green. This is smooth and little y, that's going to be smooth green. Let's see. This is, well, okay, this is smooth, and that's little y, that's going to be green. So we just got our smooth greens. You can kind of figure out the pattern, right? So these three are going to end up being Kellogg's Corn Pop looking little things, right? So those are going to be wrinkled yellow. And then lastly, we have the runt of the family, right? So we have little s, wrinkled, and little y. So that's going to be wrinkled green. And so there's your 9-3-3-1 ratio phenotypically. 9 right there, and then 3, and then 3, and then 1. And so there's that 9-3-3-1 ratio I was talking about. Again, he didn't use Punnett squares. He calculated all this stuff using probabilities. And then here's the saddest part of all of this. He had this brilliant body of work. Mendel published his results after 10 years of work. Brilliant, massive body of work. He published in what is now PNAS, the Proceedings of the National Academy of Science. And he had so much data. Remember, this would be like me, the world is round. And then this would be like me walking in and telling you, blending, people thought the world was round. It's kind of like thinking blending was so entrenched, it was entrenched as the world being round now, right? And that'd be like me coming forward and saying, okay, I know you're going to have a hard time believing this, but I have a tremendous amount of data and experimentation, and I wrote it all up, and it turns out that the shape of this planet is pyramidal, right? And so you would basically look at me like I was crazy, probably wouldn't even look at my data. If you did look at the data just because you're like, what is this guy talking about? Then there was so much of it. Because remember, he did 10 years of work and he was doing massive sample sizes. So there was just so much of it that nobody really probably looked at it thoroughly and actually was capable of understanding it all. And so Mendel published his results after 10 years of work, and then basically nothing happened. Right? He died in anonymity. Nobody actually even recognized his work until far after his death. So let's do a quick recap. Mendel had no knowledge of mitosis, no knowledge of meiosis, no knowledge of chromosomes, but by 1900, right, it became obvious that genes and chromosomes had a lot in common. Now, I took this table from a different textbook, but remember I mentioned Walter Fleming originally identified thread-like bodies that turned out to be chromatin and chromosomes. And then in, I believe it was 1904, Walter Sutton actually linked what he witnessed under a microscope with that stuff we just talked about in the cell cycle, the chromosome dance, right? So the chromosomes condensing and then lining up at the metaphase plate and then splitting apart, etc. He was like, what just happened? What was that? And he goes back to the scientific literature and he finds this work from this obscure monk from the 1800s, decades beforehand. So Mendel died in 1880, the 1880s. So, a couple decades beforehand, that just exquisitely detailed what he had just witnessed with his own eyes under a microscope. This guy basically predicted all of that using probabilities and things and said this is the only way this could all work out. And so once we get into the 1900s, it became obvious that genes and chromosomes have a lot in common. Genes, right, so in Mendel's inherited factors, occur in pairs, and he talked about alleles, and then chromosomes occur in pairs, and we talked about homologous chromosomes. Members of a gene pair separate from each other during meiosis, right? Obviously, that's not Mendel terminology, but it turns out chromosomes, right? Members of a homologous pair separate from each other during meiosis. Members of one gene pair independently assort from from other gene pairs during meiosis. members of one chromosome pair, independently assorted from other chromosome pairs during meiosis. So the foundation had been laid for basically Mendel's work to be rediscovered. And in the early 1900s, it was rediscovered. And so then people started looking at it more critically, more, well, they actually looked at all of it and tried to actually, they were motivated to understand all of this complex material. And they did. And so then they were starting to think, okay, so this is, there were still a lot of people in the blending camp that were like, well, okay, that's nice. This is how pea plants work. This is not how everything works because everything else works by blending. You found an outlier. You found something that doesn't work by blending. So, that's fine. But of course, scientists were able to say, well, okay, hold on, we should check that. And they started looking at other organisms and other aspects of genetics. And so that's when really modern genetics was born. And so now there's all sorts of other experiments being conducted in other organisms, and new things are being determined, new things are being discovered. And so genetics is now growing as a field. And so that's where we can now say, okay, well, we've covered Mendelian inheritance. Now let's look at some variations of Mendelian inheritance, because there are many. Mendel only figured out a few things. I mean, he figured out quite a bit, I should say, but he did not figure out everything. So to quick little recap, we know that genes are located on chromosomes. We know that each gene is, so we're basically translating Mendel's work to modern day, right? So we know those inherited factors are genes. Genes are located on chromosomes because the link had been made between Mendel's work and chromosomes. And so genes are units of heredity, and they are located on chromosomes. Each gene is located on a specific site on a chromosome called a locus. And so just so you know, like this is some modern information, but it'll help us in our discussion. Humans carry somewhere between 20 and 25,000 genes on their 23 pairs of chromosomes. And so there are 22 numbered chromosomes called the autosomes, and then one sex pair of chromosomes, the either XX or XY. We don't even know exactly how many genes human beings have, even though we've sequenced the human genome, because that means that depending on your tolerances of your analysis of the human genome, basically there are candidates for genes, right? We haven't checked every single one. And so there are as few as 20,000, as many as 25,000 candidates for genes. And, many of those are probably going to be genes. So we're probably going to have somewhere right around 20 to 25,000 genes in the human genome. And then we've talked about this before. Not all chromosomes have the same number of genes. Some chromosomes have more, some chromosomes have fewer. For example, I mentioned this, right? So I used to work at Lawrence Livermore National Laboratory. We were given chromosome 19 to sequence into the Human Genome Project. And, everybody wanted chromosome 1 because that's the one that was thought to have all the most genes on it because it's the biggest chromosome. And when, people at Livermore got chromosome 19, everybody was kind of disappointed and it's like, oh, sad. We have this dinky little chromosome 19. It's the 19th biggest chromosome, so it's really small. But it turns out it was luck. Chromosome 19 is the most gene dense. So while it's very small, it actually has the most gene per capita, if you will, most genes per capita, and so it's quite gene dense.